Part 2 is about DNA sequencing using a nanopore. So the title, I think, in the tutorial website was, no, in the workshop website was somewhat different, but it's basically about the same, which you will find out very soon. So it's basically molecular modeling for nanotechnology. I'm going to show this slide again to emphasize that the sequence of DNA is written in atoms. So really the difference between A, C, G, and T is just a few atoms. So the sequence of DNA is of importance to biology, and not surprisingly, it has been one of the most expensive projects done at NIH, the Human Genome Project, right? So the Human Genome Project initiated in the 90s and finished 2 years ahead of schedule. And it was also one of those projects that used less money than was allocated for it, which is very unusual. So what was the outcome of the Human Genome Project? The Human Genome Project determined the sequence of an average human being, and I'm going to show it to you now. So the sequence of DNA starts with 5 prime, and then it goes like that. Yay. So I have 3 more million pages, and then I'll be done. So it's basically very, very long, and it's just 4 letters. So in fact, although it's only 3 million pages more, it's still can fit on the CD, so you can do a simple calculation, right? So it's a 4-letter code. It's 2 bits, so it's about 750 megabytes. So why do we care about DNA sequence? So DNA sequence, well, it's not only that it's good to know, right? But there are also some practical reasons. Primarily has to do with health, right? It's also interesting to know that among unrelated individuals, we are 99.4% similar, which still leaves about 1 million differences. Just to illustrate how that would work, so here's, for example, my code, and this is Chris's. See? There's a little letter over there. That's about it. So not so much, but still a lot. So what is it? Why do we want to know human genome? And I take it back. Why do we want to know individuals' genome? That's what this is about. So we want to sequence DNA of everyone because it is important for making progress with healthcare research. So there are various applications of whole genome sequencing. It's still kind of expensive. So right now the cost is about $5,000, but still it's kind of expensive to do it routinely. But why? Let's say you can do it routinely. What would you do with that? So one thing is that you would sequence genomes just to know what are your predispositions for over 4,000 diseases. So that's good to know. Next, whole genome sequencing is really important for developing cancer therapies. You know, DNA is a disease of DNA. Basically, mutations accumulate and eventually leads to cancer. So you really need to sequence the genome. Ideally, you would sequence everything from different cancer cells to know what is the molecular origin of cancer at the DNA sequence level. And this is actually not a very easy task. As you may know, cancer is not just one disease. There are different kinds there are hundreds of types of cancer. And also within one cancer, there's also a huge heterogeneity between different cells. So really what you want to do, you want to sample maybe genomes from thousands of cells from one patient. That's just to start. And then you would want to see how cancer evolves. So you really need to have the technology for high throughput, low-cost DNA sequencing just to develop drugs. One of the applications currently of the whole genome sequencing is in this prenatal diagnostics. So that's something, I don't know if you know, you probably don't know, but say when a woman is pregnant, in the bloodstream, you can find DNA of the unborn baby. So in general, you have lots of DNA in your blood. I don't know. So I didn't know about it, but you have fragments of DNA in your bloodstream. So in principle, you can just take it from there. And if you can actually distinguish DNA from the mother and DNA from the baby, then you can basically sequence the genome of an unborn baby without doing any kind of intervention. All right. So I like showing this slide. It illustrates amazing progress of technology. This slide shows the cost of sequencing DNA versus time. So it doesn't start in 90s, unfortunately. I like to show the ones that starts at 90s, because in 90s, the cost to sequence one base pair was $10. So $10 times 3 billion base pairs is quite a big number, almost astronomical ones. And that's where the human genome got its budget for. But then, luckily, the cost of DNA sequencing was dropping really rapidly. It was following the Moore's law. So Moore's law says that the number of transistors doubles every 2 years, something like that. So, and the initial decline in the cost of whole genome sequencing was related to making things smaller, optimizing, putting everything on the same chip and stuff like that. It was dropping exponentially. Look, this is logarithmic rate. So 10 million, 100 million, 10 million, and so on. So although I was already good enough, if you extrapolate that line, you'll see that it will reach the cost of a $1,000 genome by about 2040, which was too late to wait. And that's why NIH says, all right, we'll do something unusual. We solicit proposals, all kinds of proposals, to develop different types of sequencing technologies, not incremental improvement of something, but really radical new sequencing technologies, and we will fund them. And that's what they did. In 2005, they initiated as revolutionary genome sequencing technologies. And that's an example of a problem that was solved by just putting extra money. It's amazingly, it worked. So when this program initiated in 2004, the goals were the following. They wanted to have a technology to sequence human genome for $100,000 by 2008 and by $1,000 by 2013. And that's back when the costs were already in like $20 million. So that looked really impossible to achieve, but surprisingly, they were right on target. So we reached the cost of $100,000 per genome in about 2008, and we are actually approaching a $1,000 genome mark this year. In fact, beginning of the year, one of the companies announced that they reached a $1,000 genome mark, but it really depends how you count, because you can determine the sequence to a different accuracy and so on. So I think coming up with the right number is kind of difficult at this point, but you can see that it drops faster than exponential. People sometimes say that this is one of those technologies where you see the most rapid drop of costs over time throughout the history of human civilization. All right, so how do we sequence DNA, or how was it done previously? So conventional DNA sequencing method dates back to 70s. It was invented by Sanger, and he got his second Nobel Prize for it. So basically what he did was to trick a little guy here, DNA polymerase, to read out the sequence for us. Okay? So coming back to my very first or second slide, I told you that the DNA sequence is written in atoms. So imagine if you were to build a tool that would just directly see the DNA sequence. You know, it looks like a very difficult task. But nature had billions of years to do it for us. It evolved enzymes like DNA polymerase that can recognize the proper nucleotide and incorporate it. And then what we do is we trick it so that we can read out, we can use it to read out the sequence for us. And I don't have time to explain exactly how it's done. In very briefly, what we do is we add modified nucleotides to the mixture, normal nucleotides, at the rate of 1 every 1,000. And this little guy, it can still take the nucleotides that has a fluorophore label attached to it and incorporate it into the growing strand. But once it's done, it cannot add another one. Okay? So basically, you end up with a mixture of nucleotides that have different labels at the end. And the only thing now to determine the DNA sequence is to find out the length of the fragment. And that's you do through gel or capillary electrophoresis. So you end up with a matrix like that, and then you align. So that's basically how it was done previously. And there is a limit to how far you can push this technology. First of all, you need to have labels, so they cost money. Second, you need to use this gel, and there's a physical limit of how accurate you can determine the length of DNA using a gel, and that's typically terminates at around 1,000 base pairs. So more than 1,000 base pairs, it's very difficult to distinguish if it's 1,000 or 1,001. So that was a limit. So since then, there have been several disruptive technologies, which actually why the cost of sequencing went down super exponential. It's not because there was an incremental improvements to the previous method. No, basically what the researchers did, they say, okay, we'll just do it differently, and that also came. So there are several companies involved in this business that basically built on the patents developed by different groups. But basically what they did was develop a technology where DNA is split into very short fragments, and then using very small arrays of cells to carry out many reactions in one small volume. So that's basically so-called next-generation sequencing methods. So Illumina is a company that is fairly famous for it. Ion Torrent, they engineer a very small pH meter. So basically what they have is a little bead where they add a DNA that is amplified, and then they measure the change in the pH. So that they have really, really sensitive pH meter. And but most of those second generation sequencing methods, they still use amplification to read out the DNA sequence. Meaning, so you take a sample from a patient and then you amplify the amount of DNA through polymerase chain reaction, and then you do sequencing by synthesis or something else, but you do it in a bulk, meaning you have many molecules that contribute to the signal. Ideally, you want to take one molecule of DNA and read out its sequence directly. So that was pushed forward by those two companies, Pacific Biosciences and Helicus. So Helicus went bankrupt some time ago. Pacific Biosciences are still fighting. But they were the first ones to demonstrate single molecule readout of DNA sequence by FRED, basically, fluorescent energy transfer. While that was going, there was another idea, and that's what I'm going to tell you about, the nanopore sequencing of DNA. How many of you heard about nanopore sequencing of DNA? All right, 3 people, not bad. It is a method. It was an idea. It is an idea, basically. It has been put forward 20 years ago, and in some form, it's shown here. So basically, what you need to sequence DNA using a nanopore is just a nanopore and a DNA, and a method to measure the current. So what you're looking at is a membrane channel called alpha hemolysin. It is embedded in a lipid bilayer membrane. So it's a membrane channel. It usually lives there. The membrane channel has a hole, and the hole is filled with water. So this particular membrane channel does not usually transport DNA in biological settings. It's there to punch holes. It is a toxin. But surprisingly, it's just the right size to admit single-strand DNA. So once you have DNA molecules placed in one side of the solution and you apply electric field, the DNA molecule will be threaded one by one into the nanopore and exit on the other side. So why does it happen? It happens because DNA is negatively charged. So it is a highly charged polymer, so if you apply electric field, it will move in electric field. Now, if there is only one little hole where it can pass from one side of the membrane to the other, it will do so. So here's a very old simulation that was done a long time ago that illustrates this process. You have a DNA molecule, and it passes through the nanopore like a spaghetti, I would say. It ends up on the other side. So this is a nanopore translocation, term that I will use very frequently. All right, so once you have the DNA going from one side of the membrane to the other, how do you read out the sequence? So it turns out that while DNA is passing through the pore, it will block the ionic currents that passes through the pore. So imagine everything happens in solution. So you have dissolved ion, minus and plus, all around. They're not shown here. So when you don't have the the DNA in the pore, then you basically have an open pore current. And when the DNA enters the pore, the current will be blocked, right? So that's your readout signal. So really, you're measuring electric current. It's not electrons. Well, it's ions, dissolved ions that pass through it. That's what contribute ionic current, but that's what you measure. And experimentally, it was found that those blockade level kind of depend on what kind of DNA is in the pore. So an ideal case scenario would be this one, where you have a signal like that, open pore current, and then it drops, and then you have this stepwise pattern, which will tell you the sequence of the DNA. So you just look at the magnitude of the current, and that's it. So in principle, this is really revolutionary in that sense that you don't need to label DNA. You can use very long fragments. In principle, there is no limit for how long the fragment could be, as long as it finds the pore. And you're reading out electric currents, so in principle, it could be very, very cheap, very fast, and also a very scalable technology. So it's been proposed some time ago, but only now we are getting to the point where we can actually say, yes, it's working. So let me show you this movie. This actually illustrates the readout process. So you're looking at the DNA confined in this alpha hemolysin, and those balls are the ions that are going up and down through the pore. You can also see the problem with this method. In this particular pore, you have about 12 nucleotides in the constriction. So the ionic current is kind of convoluted over the entire length of the pore. So basically, this was never realized with alpha hemolysin. And all of those dependence of the ionic current on the sequence were done for DNA made entirely of A, C, G, or T nucleotides. So it used to be that when I was showing this slide, people would laugh, but now, finally, my collaborators at University of Washington, they've actually experimentally demonstrated DNA sequencing using a nanopore. The key to their success was in using a different pore called MSPA. It has a very nice shape. It looks like it was evolved to sequence DNA. Of course not. It was evolved to do something else, like maybe import glucose, or I think that's what it's doing. It's called MSPA because it's Mycoplasma porin A. So it's basically one of the porins that is in the outer wall of the bacterium. But it has this very nice shape that can accommodate just 2 or 3 nucleotides. And the other key thing was in using a motor enzyme over here to slow down the DNA translocation. So in this particular case, you have electric field. So electric field points up. DNA is negatively charged, meaning the force of the electric field pulls it down. And typically, if that's it, then the DNA passes through the pore very rapidly, and basically you're not able to register signals from individual nucleotides because it passes too rapidly, basically. So what they've done, they've added this enzyme. It is again DNA polymerase, But it's not used to, we are not using DNA polymerase to read out the sequence. We're only using it to slow down the DNA motion. So what it happens, the DNA polymerase synthesizes a complementary strand, pushing the DNA up. All right. So in a synthesis mode, without electric field, it would push it up. So you have a tug of war here. DNA motor pushes it up, and electric field pushes it down. And that allows slow down the process of DNA transport through this very little constriction considerably, so that you can resolve those single nucleotide steps. And that's basically what is. Those are the raw data from the paper. You see there's the steps. They're well defined, and but they're also and they're also quite big. Then what you can do, you can use a separate set of measurements where you calibrate your apparatus, and you can interpret this ionic current recording as a sequence. Okay, so that was done and published in 2012, and was probably the first demonstration of nanopore sequencing that worked.
Is it well known the forces between two that's interacting on the DNA strand?
You're asking what is the force that is?
Yeah, I'm asking if the forces are known in the tension.
The magnitude of the forces, yeah, it's about. It varies between 10 and 30 piconewtons, roughly. Although it's actually a very difficult thing to measure experimentally, much easier to do a simulation, which we've done. Yeah. So about the same time, Oxford Nanopore Technologies announced this product, which looks like that, called MinIon. It goes into your Mac, and here you put your drop of blood, maybe, I don't know. But basically, they said that they will have this product last year, but unfortunately, it's still not out. They are in beta testing right now. So I should give you disclaimers. I actually found the project in my group, so this is not advertisement, okay? But they announced, that was really a big deal. So they announced this method, the device, the first commercial application of nanopores. And those are the specifications. Read length up to 100,000 base pairs, which is really shocking. And it comes with accuracy problem, so it's not super accurate, 96%. So in that sense, it's a trade-off. You can get a very long fragment, but at the limited accuracy. It's probably, if you supplement with another sequencing method, then you're basically winning all the time. I actually don't know, honestly, what kind of pore is in there and what kind of enzyme. This is proprietary. It's definitely not the one that was shown by UW people. But there are many proteins. There are many DNA processing proteins that go along the DNA and unzip it, like helicases and so on. And with 100 people, you can really try all mutations and find the one that works. So there's one product here, potentially, maybe. This is another one that came very recently. I was kind of shocked to see the prototype. This is not a coffee machine. This is actually a quantum, for the first quantum sequencing device. It's made by a company, this Quantum Biosystems in Japan. So what's in there? I think this is in there. Let me explain what that is. You're looking at a cartoon, first of all. This is a cartoon showing an electrode. This is a gold electrode. And the spacing between the two gold electrodes is a few nanometers, so really small. So the key here is that you have this spacing that is comparable to the size of the nucleotide. So the DNA is squeezed through it. There's some very fancy movies showing how it's done, how it's unzipped, and so on. But the key step happens here. Now you have a difference in electric potentials, and electrons would jump from one electrode to the other through the DNA. So you're measuring tunneling current. So you don't have a contact. So it's like a gap, right? So if you crank it up on a microscopic scale, you would see a spark, which probably why you see this. But of course, at the nanoscale, you just have quantum tunneling. And once you place the DNA there, your current increases in a way that is specific to the DNA sequence. And the company has shown some data where they sequence short fragments of DNA, but that's, if it's true, this is really a big deal because it's the first time that I know that quantum mechanics is used for bio applications directly. And so on. So there are actually many companies that are interested in this subject. So pretty much every big company that has a nanofabrication facility is interested. And the reason is, well, the market for chips kind of saturated, but the bio market is going to grow and grow and grow. The market for sequencing DNA is estimated to grow exponentially for the foreseeable future. You can imagine that you would want to sequence anything. Like you go to a shop and you want to know what you're buying, you know, just have your handheld reader and so on. So people will be sequencing everything, and this is a big niche for them. And they have expensive nanofab facilities, and finally there is an application where they can use it. So making those tiny pores is expensive, and not that many places on Earth exist where you can make them. So that's why. All right, so what am I doing with this? This is a modeling workshop. So I'm going to show you how we use molecular dynamics simulations to help our experimental colleagues to design better nanopore readers. And there is another, so today you'll have two tutorials. One is on modeling DNA protein system, and the other one is on modeling nanopore systems. So both of them are kind of long. So you can choose one and just work on it for the evening, or you can try one and then the other. It's up to you. But that's basically what they're about. So let me give you some examples. So here's one, the first one, coming back to the MSPA system. So how do we know that different DNA sequences produce different ionic currents? So what you can do, you can add a big protein to the end of the DNA. This is a streptavidin, and repeat experiment. Because you have a streptavidin attached, it will block the DNA permeations through the MSPA like this. And that will give you a really long time to accurately determine the identity of the nucleotide that sits here. So those are the results of the experiment. So you are looking at ionic current blockade versus DNA sequence. And you can see that they are separated by a large margin. But the most interesting part is that the nucleotides that are smaller pyrimidines, they block the current more than the nucleotides that are bigger. So that's kind of completely counterintuitive. Furthermore, if you know the blockades for A, C, G, and T individually, you cannot predict what the blockade for a triplet would be. They basically don't add up. So the question was, you know, what's going on in there, and how, like, why do we have this differentiation? Basically, we could have a device, working device, but still don't know why it works. And of course, we couldn't live with that, so we had to simulate it, which is what we did. So how do we go about simulating systems like that? It's really like cooking. You need to find a recipe, so you go to Protein Data Bank and you Google for, well, you find your structure. So in this case, you type in hemolysin, and you'll get an X-ray structure. So I think this step everybody knows how to do. And then, well, it's just like cooking. You add the DNA. I guess that's not the most common process. But the rest is, so you add a little bit of fat, so that's your lipid bilayer, a little bit of salt, and water, and end up with a system. So that's your all-atom system where everything is assembled. So the next step is what? Turning the gas on, right? So you give a little kick to every atom, turn the temperature to 300 K, and F equals ma many times for every atom until it's done. So how do we compute ionic currents? So that's basically the first task that we had to deal with in this project. It turns out to be extremely easy. and accurate. And that was shocking, actually, for me, because it was right on the target. So what we do is we just apply constant electric field in the direction normal to the membrane, like that. And that constant electric field, it will interact with every charged particle in our simulation system, which is basically every atom, redistributing charges in the system. So you have an applied field, and then there will be a reaction field from the system. So that reaction field plus the applied field will produce a very complicated pattern of the electrostatic potential in the system. Here it is. So you see it's uniform away from the pore, and there is a gradient of the electrostatic potential through the pore. So that is the gradient that drives ionic current through. So in a way, you can think about it as imaging the electrostatic potential in your system by means of molecular dynamics simulation. And this is applicable not just to ionic current simulation, but basically to any simulation that you've ever run. With all atom, you know the electrostatic potential already because you need to compute long-range interactions through the PMF map, right? So the only trick was to extract it from all atom MD, and that's what we did. And this is also part of the tutorial, explains how to do it, how to basically get your electrostatic potential from your simulation. But because we have we know position of every atom at every time step, we can easily compute the ionic current. So you see those balls of different colors, they move up and down. That's because electric field applies, and then we can trivially compute the ionic current. It's just the displacement. So that's the change in the coordinate of the ion times its charge over the time, sum over all ions, and you get the ionic current. If you're wondering why this movie is so blurry, that's because I updated to Keynote. And it's just, for some reason, all older movies just don't look any good anymore. And it's only one way, so I cannot actually go back. All right, so that's what you get. When you compute ionic current, you'll get a mess like that. So it's really fluctuating a lot because the motion of ions is stochastic. If you could have an amplifier that could record with that bandwidth of nanoseconds, you would actually record something like that. But most of the time, ions just move up and down. So it is a kind of a noise that can be filtered out. So one practical way to filter it out is just to integrate it. If you integrate this noisy curve, you'll get something that looks much nicer, straight line, which kind of tells you that the simulation reached a steady state. So what it is actually is the number of charges, elementary charges that passes through the pore. Typically, once you get 50, you can approximate the current by taking a slope of this line. And so the slope is the current. And then you can do it at different voltages, and you can get current voltage relationship of an ion channel directly from a atom MD. So it is very straightforward simulation, and it is described in the tutorial. It works very well, surprisingly. For example, in this case, experimentally, we should get 150 picoamps, and we got 130 picoamps from our simulation just like that without doing anything, right? If we change the polarity of the bias, we'll see that the ion, some slight rectification of the current, just like an experiment. So, so in essence, it's one of those few problems that where we can compute a value that is directly comparable to experiment, ionic current. And the reason for that is that the permeation of ions happens at the nanosecond time scale. So if you model it for a long, if you run the simulation for a long time, you get lots of statistics, you can predict what the error bars are, and it works very well. All right. Coming back to this question, so why do we have different different ionic current blockades for different sequences? So to answer this question, we actually had to go to a special purpose supercomputer, Anton. So we soon find that if we just do this big simulation with everything present, the ionic current fluctuates so much that we basically would not get any sequence-specific currents at the time scale of our simulations, like typical time scale, like hundreds of nanoseconds. So we have to go to Anton, and everybody knows what Anton is? Anton is this supercomputer that is at Pittsburgh. Well, it was developed by DSO, but the one that we use is actually here. And so Anton has this problem. It cannot handle big systems, so we had to cut off a piece of a channel. But anyway, so here's the setup of the simulation that we did. We cut off this piece of a channel, and of course, before going with expensive simulation, we made sure that the electrostatic potential from a small part is the same as from a big part. And then we added a harmonic spring here to hold one atom here. And that achieved two purposes. One, we fix the DNA. I mean, we fix this atom in the DNA so it's not transporting through the pore. And the second, we actually measure the force that is applied by the electric field in there. And that's it. So we had to constrain certain atoms in here so that the channel doesn't fall apart. And so here's some results. So you're looking at this dance of different sequences. This is poly A, this is poly T, I believe. And you can see that the DNA, single-strand DNA, just moves around a bit. So that was, for experimentalists, that was really revealing because they didn't know that there's so much conformational fluctuations in the system. Now, if you look at the ionic current as a function of time, you could see that we no longer have this very rapid stochastic motion because we already filtered out. So every point here is 100 nanoseconds. So this is only 10 microseconds. Now we have simulations that go up to 50 microseconds. But basically, you can see there is long, slow fluctuations in the current, and that has to do with the change in the DNA conformations. So can our simulations reproduce experiments? That was the first question that we've asked. And if you look at the distribution of the ionic currents, I would say that yes, they can. So just like an experiment, we see that the smaller DNA nucleotides block the current more than the larger DNA nucleotides. And if you ask the question, what is the molecular reason for it, we have the answer. The answer is basically everything is determined by the amount of water molecules that are in the pore. So what you're looking at are water molecules. And there are water molecules that are structural water molecules, the ones that are near the surface of the pore, and the ones that are bulk-like. And basically, if you just count how many water molecules are in the constriction of the pore, you can tell what the current is. So for example, if I plot here the ionic current and the number of unstructured water molecules, you can see a one-to-one correlation between them for every system that we've studied. So basically, two messages from this. One, indeed, we can reproduce experimental system where we measure the current through the nanopore. And second, our simulations not only reproduce experiment, but also tell something new to experimentalists. What is the molecular origin of ionic current blockades? And here in this case, R is just the number of water molecule that is in the constriction. Are there any questions to this part? All right. OK, so the Nanopore tutorial has several parts, and they can be done independently. The first part is just the DNA. How do you model DNA? And we teach you all about it. Where do you get the DNA structure, how you put it in solution, and so on. Then there are options. Option number one is to go and see at modeling systems where you have a biological pore in a membrane. That's option number one. Option number two is to look at synthetic system, which is the rest of my talk is about. In general, there are many nanopores, and we worked on all of them. There are nanopores in polymer films, If you are interested to model them, let me know. Nanopores in lipid bilayers, the ones that I've just covered. And then there are also those called solid-state nanopores. They are made in a solid-state membrane, like silicon. There are also active structures. By active structures, I mean that they have they're not made in insulating membranes. They're actually made in metals or semiconductors. So it's a very, very rich field in the sense that you can combine different physical mechanisms with biomolecules. So I find it particularly exciting because I can use my physics expertise and also my biology expertise, and this is really the confluence where lots of interesting things can happen. So in the case with solid-state nanopore, it's just like with biological pores. So the membrane is now has a more regular shape. Typically, it's a double cone. If you wonder why it's a double cone, if you ever saw in a movie people shoot through the wall, then it's usually a double cone because you get some material extracted from the pore while you drill it by shining electron beam, and the material comes out on both sides. So if you take a DNA molecule and place it near the pore, apply electric field, it will get in and out on the other side, and then the next one will come, and the process will repeat. Here's an experimental trace. Open pole current, DNA in, open pole current, DNA, and so on. The problem with solid-state nanopore is that it's typically much thicker than the single base. So the question was, how can we use a system to sequence DNA if we cannot confine just a single base in the pore? And the answer to this question was, of course, the Scotch tape, right? That's the answer. You are all familiar with the Nobel Prize that was given in 2010 for the discovery of the graphene and demonstration of its use. And of course, the graphene is a perfect material for nanopore sequencing because it's atomically thin. So here's the proposal. So we'll take a graphene, layer of graphene, with a single pore in it, and maybe we can use the graphene to sequence DNA. And here we have several options. Option number one, we can just say, well, let's pass it through the pore and measure the ionic current, just like we've done with biological enzyme. Option number two is to actually use unique electronic properties of graphene to do something more clever. For example, you can create a nano gap using graphene and measure tunneling current, or you can create a nano ribbon and just measuring a transverse conductance through the graphene and see how electrons and holes scatter around the edges with and without the DNA. So there are many avenues to reading out DNA sequence using graphene. It's a very hot area of research, and it actually turns out to be quite easy to model. I don't think we have graphene in the tutorial that we are presenting to you. Modeling, though, would be very similar to modeling silicon oxide, and if you want to try it today, you can do it. I think Jim can help you with that because he's working actually on graphene nanopore project for his science project. So what happens when DNA encounters graphene? So here's an example. We typically start our simulations with having one end of the DNA already introduced in the pore. Why? Well, because we don't want to wait for it to find the pore, and that's the process that is kind of long and tedious, and we want to accelerate the simulations. So the first observation was that the DNA adheres to the surface of the graphene very rapidly in a specific way. You see, it kind of goes flat and kind of gets stuck to it. But once it's stuck to it, it still keeps on going. You know, it moves around, rapidly exploring the vicinity of the pore. Why does it happen? Why does it move around like that? The answer to that is because graphene is atomically smooth. So every piece of graphene is just like the other one. So there is nothing preventing the DNA from moving around it. So it's basically a 2D diffusion without almost zero friction. That's why it moves around so much. So that was the first observation. The second observation was that the DNA actually moves through the pore in steps. So if you look closely how it actually transports through the pore, you'll see that it changes in discrete steps. So here's the number of nucleotides permeated, and you see that it goes like one after another, which is actually excellent for DNA sequencing because you want to have one DNA at a time in the pore, changing rapidly. So basically DNA, the graphene itself acts as this polymerase enzyme that was used for biological pore to control DNA transport. But here we can just let the system do its physics and see how the process of unbinding on one side and rebinding on the other side produces stepwise motion. So one other conclusion from the molecular dynamics simulation was that you actually don't want to use a single layer graphene nanopore. So there was lots of speculation that that would be the optimal material. But if you use only one single layer, basically you end up never having a DNA in the pore. So if you want to sequence it by measuring ionic current passing through the pore, that's not the best choice because you don't have anything of sequence to modulate your current. So ideally you would use a 3-layer pore. And right now it's possible to actually tune your experimental system to choose which one you want. We have also found that a method to control the DNA motions through the pore. So I'm going to skip through this. So, you know, the graphene is a conductor. So in principle, we should be able to charge it by applying a bias to it. So here's the system we considered. So one end of the system is grounded. The other end is applied is held constant at a certain potential. So that's just like before. But we also have a different potential that we apply to the membrane, which induces overcharging of the graphene. So we can change this potential to have graphene positively charged or negatively charged. All right. How do we do it in practice? In practice, what we do, we just assign charge to the graphene and then compute the potential that corresponds to it. It's kind of difficult to do it other way around. It involves electronic structure calculation. And that amazingly gave us a way to control the DNA transport with high precision. So what we found was that depending on the charge of the graphene, the velocity of the DNA can change dramatically. So if we charge graphene negatively, the DNA basically doesn't move, but if we charge it positively, it moves very fast. So what we could do, we propose so-called stop-and-go mechanism for DNA transport, which I will illustrate using this movie. Again, if you want to know how to make those movies, ask Chris. So what you're looking at here is a graphene, and there's a gradient in the background. So the gradient in the background shows you which way the potential points. So it's 500 millivolts, so the DNA would want to move down. Eventually, I will run out of DNA and switch the potential to point up, so it would go the other direction. Okay, so just telling you what happens. So the membrane is initially positively charged. So when it's positively charged, according to our hypothesis, the DNA would actually want to go down. And it happens. Now we want to stop it. So to stop it, we are not going to switch the field. We're only going to charge the membrane negatively. So the field still pushes it down, but the motion is arrested. Now we are going to switch the direction of the gradient. So now it wants to go up, but it can't because it's pinned by this potential. Now we let it go, and now we're going to catch it again by switching it to negative value. All right, so we got it, and now we will do it again. It wants to go down, it doesn't. Charge it positively, and it goes, and now we can catch it. So let's kind of illustrate where I'm going at. So what I like about the molecular dynamics simulations and the resources that we have right now is that you can do this not just once, not twice, but you can do it 5 times, 10 times. So kind of getting something to the point that your experimental colleagues would actually take it very seriously and would invest lots of resources trying to verify and realize your proposal. Okay, we have a few minutes left, and I really don't want to overburden you with different details. I just want to tell you, broadly speaking, what kind of other processes can we study. But before I do that, are there any questions to the previous graphene part? Yes?
Yeah. Why when the graphene is charged positively and negatively, the DNA is still attached in the previous slide?
Right. So that's actually an excellent point, and we have a paper on that. Something very interesting happens to DNA when you charge the graphene. Because DNA is negatively charged, when you charge the graphene negatively, it would actually unbind. But there is a critical charge at which it happens. So at some level of the charge, it would still remain bound, but not so strongly. But it will bind stronger to the pore edge. When we charge the graphene positively, what happens to the DNA, it changes its conformation depending on its sequence. It's very, very interesting because you have a partial distribution on the base and on the backbone that is sequence specific. For example, for one sequencing, what we see is, so I don't have a slide here, unfortunately, so I'll have to show it like that. So this is a base, and what it does, it tilts, right? For one sequence. For the other sequence, it kind of stands up and does like a ballet, like that. The other sequence, it kind of turns up, and the base comes off. So it's actually very rich behavior. I just don't have time to actually describe what happens. But it's a particular system because you have a charge here. The DNA is also charged, but you have two other effects. One is hydrophobic interactions. So the base is hydrophobic, graphene is hydrophobic, so it sticks to it. And the final one, everything happens in 1 molar KCl. So the charge is screened. So 1 nanometer away, the DNA doesn't feel the charge anymore of the graphene. So it's this very thin layer where the charge can affect, and the particular distribution of charges at the DNA matters. So that's what happens there. Yeah? Yeah, so we chose as, so usually what we do, we put a DNA a little bit halfway through the pore, and then we equilibrate it for 100 nanoseconds, and by that time it was completely attached to it. Then what we did, we fixed DNA, changed the charges on the graphene, equilibrated it for like half a nanosecond to let it react, and then let go. Okay. All right, a few other systems to explore with solid-state nanopores that I found of interest. So we've talked about this sequencing DNA by tunneling, right? That was this quantum biosystem system where you have electrons hopping from one side of the membrane to the other. But typically, DNA is shown here as a straight helix, but it's not the case. In fact, this is what the DNA looks like. This is an all-atom simulation, it's fairly long. It's just like it's just a polymer, right? It doesn't have any structure, a single strand. So we used molecular dynamics simulation to discover a method to control the conformation of the DNA, and it was through temperature. So you might be familiar with nanoplasmonics. So basically, if you take a small pieces of gold or other method, and they are of a right shape, you can shine laser, and the laser will interact with electrons in the metallic structure. And several things can happen. The simplest one is that you will excite those plasmonic excitation, and they will rapidly heat the particle. It was shown that you can heat like small particles to temperature. You can actually melt them in solution. So you have particles in water, you can shine laser on it, and they will melt and still be in solution. And then you could switch it off, and basically within a few nanoseconds they will solidify. Because they're so small, this process of heating and cooling is very rapid. And you can do it with light, and that's great because light you can modulate very fast. You don't have any capacitance problems that you have with the electric field. So those are two groups that published in 2013 on this plasmonic heating with nanopores. We basically did the same independently. We considered a system where we had a nanopore and a little plasmonic heater here. Now what we had to do, so this looks all very nice, but to do it on atom molecular dynamics simulations, what we had to do, we had to introduce multiple thermostats in our model and control temperature in different regions separately. And we have done it with NAMD, and if you're interested, again, exploring it, I don't know, for whatever system, just let me know. It's fairly advanced, but we can help you. So what we found was that if we hit the nanopores, the DNA basically will expand. And if we switch off the heating, it will contract. So basically, we can regulate the conformation of the DNA by switching on and off the heating source. So I would say it's like a kind of a molecular gymnastics of DNA. And this just stretches, contracts, stretches, contracts. And that is useful because when you want to sequence DNA by measuring currents through it, you probably want to have it in extended form rather than forming a clamp, because otherwise you would get signal from all of the nucleotides that are in the pore. So that's example number one. Example number two is this one. It's in a way similar system, but now we have two gold bow ties and a nanopore. So what it is, it's called a nanoplasmonic tweezers. Most of you probably never heard of it. It's actually very similar in spirit to optical trap setup, but at the nanoscale level. So in optical traps, you take a laser beam, and the laser beam is focused to a micron spot. And if you have a bead in there, it will apply a restraining force. Now, everybody knows that the wavelength of light is approximately 500 nanometers. And for that reason, it's almost impossible to use light to trap individual molecules, of course, unless you use nanoplasmonics. So what you're looking at here is those bow ties, and they serve as antennas. So you have a laser beam coming in. Laser beam will induce oscillations of the electrons, and if your geometry is right, and to figure out what the right geometry is, you have to solve Maxwell's equations. Then those electrons will focus the light to a nanometer spot. Once they will focus the light to a nanometer spot, you can use it to trap biomolecules directly without any kind of beads or tethers attached. Okay? So that's the systems that we wanted to study for DNA sequencing. Here's, by the way, experiment. It's done in at Delft Technical University in Netherlands. So you can see a pore drilled through two gold bow ties. This is like a miracle of engineering. But basically, we wanted to see if we can use plasmonic field to trap DNA. And again, this is one of those projects that really push the envelope of molecular dynamics simulations. Because what we had to do, we had to solve the Maxwell's equation and then somehow combine it with all-atom simulation. Here's an illustration of a system. It's cut off here so that you can see what's in there, but there are two gold bow ties. There's this density over here that represents the plasmonic field that we take from our continuum optics solver, and a DNA molecule. And how do we couple plasmonic field with DNA? So we have put in NAMD some time ago a module called grid forces. So you can define an arbitrary potential and apply that potential to any subset of atoms in molecular dynamics simulation. And that's what we use. So it's very useful if you have some kind of a multi-scale problem or multi-physics problem where you solve one kind of physics equation to get a field and then apply it to our all-atom simulation. So that field acts on DNA. And the way it does it, it traps it. So in this particular movie, you see that the DNA is attracted to the edge of the bow ties. Those errors shows you the direction and the magnitude of the local plasmonic forces. So it will eventually be trapped on the other side here. Yeah, again, to make this movie, we had to use scripts, custom graphics, so you can basically draw those errors over there. So basically, we've shown that we can actually trap it. And we were also shown that we can step it through the pore by switching off and on the electric field. So what I'm showing here is just one of the examples of multi-scale approach where you can take something that you compute with one solver and use it as an input for the all-atom MD. Let me finish with the following last system. How many of you are familiar with DNA origamis? Some of you are. So we have recently figured out how to simulate them. So what are DNA origami? DNA origami are those nanoscale systems made by folding a long strand of viral genome into basically arbitrary shape. So right now you can make all kinds of three-dimensional shapes, but no one has ever simulated them. So we have recently published a paper where we describe a method to simulate DNA origami structures with all-atom resolution. We are also in the process of writing a tutorial on that subject, so if you're interested in modeling them, let me know. We will probably have it within the next months at most. We are almost done with that. But basically, what our tutorial will do is it will guide you from designing the DNA origami using a DNA design software called CAD Nano to actually having an all-atom setup for it that you can run. And our projects in this area are focused on studying the ionic permeability of DNA origami structures. So eventually we want to use it as a kind of a scaffold on top of the solid-state nanopore. So why would you want to do that? You know, with DNA origami, it basically provides you a lattice, and each turn of the DNA has its address. So in principle, you can embed on top of this nanopore any kind of molecule or label that you want. You just need to conjugate it to a piece of DNA with the right sequence, and it will find its way in this assembled structure. So you could imagine putting like a little factory here. You can add maybe a golden nanoparticle to enhance your fluorescence signal. You can add DNA or protein processing enzyme somewhere and so on. So it's kind of at this stage, exploring the feasibility. Of course, we have to make sure that the DNA doesn't leak ions too much, or if it leaks ions, it doesn't do it in a very noisy way. But this is basically work in progress. Here we want to design. We can use DNA origami and molecular dynamics simulation to design better lattice that is ion-proof. Finally, we are also interested in exotic systems like DNA ion channels. So you can make ion channels of DNA, surprisingly. You just need to use proper attachment points, and then they will just go and embed into lipid bilayers or even live cells. And because you are designing the DNA sequence, you kind of program ion specificity of your channel just directly from your design. So from a synthetic biology point of view, this is kind of interesting. From a point of view of molecular simulation, it's also interesting because I don't think anyone ever put the DNA. Well, there are some publications, but basically interface between DNA and lipid bilayer is kind of unexplored. All right, I am done here, and I wonder if you still have any questions left for me. Yes?
You put an electric field that has one direction across the nanopore. How do you keep the ion concentrations from simply, like a charge differential from building up? Do you have a periodic boundary condition? Okay, so the electric field doesn't oscillate. It is just one direction.
Yeah, it's surprising that it works, actually. At first, actually, with this case, I first did it and then saw that it works and then realized that why it works. So basically, you have a periodic system, right? So what you have is an infinite array of membranes, and your potential drops from plus infinity to minus infinity, but your drop per membrane is fixed. Okay? And you have a continuous ionic current through all of them. So that's what it is. Yeah. Any other questions? If not, then I think our lunch is ready. All right.